Dear students, please note down. These notes are available in the description of video. Please download your notes. Dear students, note down. In today's lecture, we will discuss lysosomes. This is a topic of cell biology. In our last lectures, we have studied nucleus, and today we will clear lysosomes. Note down, these are suicidal bags. These are intracellular digestive vesicles. Or note down, these are polymorphic organelles. Note down, today's lecture very important for all competitive exams plus BSc first year students. Very important topic for final exam BSc students. Lysosomes, suicidal bags. These are unit membrane bound cell organelles. Note down, these small vesicle-like structure, vacuole-like structures which are bounded by a unit membrane, single membrane. And these cell organelles first discovered by a Belgian scientist, Christian de Douai, and the term lysosome. This term also coined by this scientist de Douai in 1955. That was an accidental discovery. That was a serendipity, by chance discovery during cell fractionation of liver cells. This Belgian scientist Christian de Douai discovered these cell organelles, lysosomes. This diagram shows different types of lysosomes and their functions. Rough endoplasmic reticulum having ribosomes, ATS ribosomes. Smooth endoplasmic reticulum without any ribosomes. And these smooth endoplasmic reticulum gives rise Golgi body, Golgi complex. And these Golgi complex produce lysosomes. These small spherical structures which are bounded by a unit membrane. Note down, the nucleus bounded by double nuclear envelope, two membranous coat. But here only unit membrane, single membrane is there over lysosomes. In these lysosomes, about 50 different types of acid hydrolase enzymes are present, and these acid hydrolase enzymes are present in the form of small granules. Here the pH in these lysosomes, that is about 4 to 5, and the pH in lysosomes which is acidic, that pH is maintained by proton gradient or influx of H ions. Note down, these lysosomes having about 50 different types of acid hydrolase enzymes, because these enzymes act in acidic medium in presence of water. These are named acid hydrolases. So many enzymes like nucleases, like proteases, peptidases, lipases, dear students, phosphatases, sulfatases, and amylases. These acid hydrolase enzymes which are involved in intracellular as well as extracellular digestion, and these enzymes which are very harmful, it means this cell membrane of lysosome which is protected by the action of these acid hydrolases due to high glycosylation of cell membrane protein molecules and lipid molecules. It means the action of these acid hydrolase enzymes over this cell membrane due to presence of high glycosylation of cell membrane protein and lipid molecules. Note down, all NEET students, this cell membrane, which is very sensitive to carcinogens, to UV radiations, to X-rays, chemicals, drugs. It means the action of these agencies like carcinogens, like drugs, like radiations, that action is prevented by cortisol, cholesterol, cortisone. Such type of substances protect this cell membrane from carcinogens, from radiations. The lysosomes, they show polymorphism. No doubt, they show polymorphism. It means the lysosomes appear in several forms. These are of following types. Primary lysosomes. Note down the primary lysosomes. The newly formed lysosome, which are derived from Golgi body. These newly synthesized lysosomes from Golgi apparatus, Golgi body or Golgi complex, having only acid hydrolase enzymes in the form of small granules, and these are bounded by unit membrane. These are named primary lysosomes. Note down the newly synthesized lysosomes which are derived from Golgi apparatus, which are bounded by unit membrane and having only acid hydrolase enzymes in the form of small granules. These are primary lysosomes. The secondary lysosome. And these secondary lysosomes are formed by the fusion of either pinocytic vesicle, and this vesicle having a fluid droplet, water droplet, liquid is there. That's why it is pinosome. A vesicle, a vacuole having fluid. Or this secondary lysosome may be formed by the fusion of phagocytic vesicle or phagosome having solid food material. Means primary lysosome, that is newly formed lysosome, and secondary lysosome is formed by the fusion of either pinosome or phagosome. That's why these are named heterophagosomes. Secondary lysosomes, heterophagosome or digestive vesicles, because in these secondary lysosomes solid food particle is there. Third one is residual body, tertiary lysosome. In secondary lysosome, the action of acid hydrolases which digest the food substances. It means in this residual body, in this tertiary lysosome, undigested food, indigestible substances are there. Primary lysosome is newly formed having only acid hydrolase enzyme. This secondary lysosome, which is formed by the fusion of this pinosome or phagosome having food material. These food substances are digested, but some indigestible material is there. Then this body having some residues, indigested food substances, then it is tertiary lysosome. And these waste substances are removed. This vesicle fused with the cell membrane and the solid waste is removed out. This process is exocytosis. Dear student, intake of water, that is pinocytosis. Fluid is taken inside. Or intake of solid food, that is salivating or it is phagocytic cytosis. And the removal of solid waste through this vesicle, vacuole or lysosome, then it is ephagi, solid waste is removed. Or it is cell vomiting, or this process is exocytosis. Exo, outside, cyto, cell, cis, the phenomena. The removal of solid waste. Intake of fluid, that is pinocytosis. Intake of solid, salivating, that is phagocytosis. And the removal of this waste, then it is exocytosis, ephagi or cell vomiting. This is fourth type of lysosome. Note down each and every point, very important. Autophagic vesicle. It is autolysosome. It is recycling center, or it is disposal bag, because this autophagic vesicle or lysosome having worn out, dead or degenerate cell organelles like mitochondria, Golgi body, ribosomes, etc. That's why these are named suicidal bags, autophagosomes. These lysosomes perform different functions. The functions very important for all competitive exams. Functions of lysosomes. First, intracellular digestion. As in these lysosomes, the acid hydrolase enzymes which act in acidic medium involved in the digestion of all types of food. Carbohydrates, nucleic acid, proteins, carbohydrates, fats, as well as extracellular digestion. When these lysosomes secrete these enzymes outside the cell, means intracellular as well as extracellular, both kinds of digestion played by lysosomes. Body defense. Note down the lysosomes, particularly the lysosomes of WBC leukocytes. And the lysosomes of WBCs or leukocytes destroy the toxins, harmful substances, means the lysosomes are involved in body defense. Fourth is very important point, mobilization of reserve food. Note down mobilization of, solubilization of reserve food, particularly dear students at the time of starvation, during fasting. In our body, in our cells, the stored food gets mobilized. It means at the time of fasting, starvation, body gets nourishment with the help of lysosomes. At the time of seed germination, embryo receives nourishment through the lysosome, because the lysosomes mobilize the reserve, the stored food. Intracellular scavenging means inside the cells, the lysosomes remove the old, the dead cell organelles, the degenerate organelles. Next point is metamorphosis. Very important. It means lysosomes play an important role during development, embryonic development. As in case of tadpole, larva of frog, the tail, the gills, the gill slits, the gills, tail, etc. All these body organs are destroyed by lysosomes. So the lysosome play vital role in the development of embryos. Next point is sperm lysine. Point number 7, that is sperm lysine. It means at the time of fertilization, when a sperm enters, the sperm fuses with egg membrane, egg coats are dissolved by sperm lysine, and the sperm lysine are produced by the lysosomes. Next point is breakdown of aging and dead cells. Next is formation of thyroxine. It means our thyroid gland. In this thyroid gland, thyroxine hormone is produced by the action of lysosomes, which causes hydrolysis of thyroglobulin. Cell division. It means at the time of cell division, at the time of mitosis, the lysosomes break down the substances which inhibit cell division. Next point is mutation. It means the lysosomes also causes, the lysosome causes changes in genetic material, chromosomal breakdown. It means the lysosome may lead blood cancer. removal of carcinogens. Such type of harmful substances are removed by lysosomes. IUD, intrauterine devices, means such substances which are such type of devices which are used to prevent pregnancy, to contraception. And these IUDs, intraceptive devices which stimulate the uterine membrane. That is, dear students, endometrium, the inner wall of uterus, endometrium, which gets stimulated by these IUDs, intrauterine devices. And these devices stimulate the endometrium of uterus. As a result, lysosomes are released. And these lysosomes prevent implantation, fixation of blastocyst. That's why these IUDs are used, in which particularly the lysosome, and these lysosomes prevent implantation. Next point is osteogenesis, means formation of bones. At the time of bone formation, some material gets removed, destroyed, like cartilage, and that cartilage is replaced by bony structure. It means the lysosomes play so many roles during development, in the removal of dead, worn out, degenerate organelles, in the digestion of food substances, dear students, and in body defense. And these lysosomes show polymorphism, having primary lysosomes, secondary, tertiary, and autophagic lysosomes. In tomorrow's lecture, we will discuss mitochondria.